Okay, I think we can commence.
Okay.
So to begin with, I would like to essentially thank everybody for attending. This is a very niche area. I know a lot of people are not very familiar with it, but there's a lot of interest going around. So what essentially gave way to this webinar. I would also like to thank the professor Theodore Abadzopoulos, who sadly couldn't be here, the great facilitator, as I like to call him, always supporting colleagues for all sorts of endeavors. And essentially, for personal issues, he just could not attend, but he sends his best. I would also like to thank Frank Deschandeau. I don't speak French. I don't know if I'm pronouncing it correctly. He's the photographer that allowed me to use the venomous snake for the poster, and a lot of people have emailed me for that specific snake. It's a Tropidolaemus sabanulatus from Borneo, and it's a male, which is why it has the red stripe along its side. So yeah, I would also like to thank David Worrell for being here with us, John McCafferty at the same time, and of course you, Senor Gutierrez. We're very glad to have you with us. Thank you for taking the time and the energy essentially to, yes, just go through this for us. So thank you very much.
Thank you very much, Thomas. It is my real pleasure to be here. Hello to David and John, and greetings to everybody. It is my pleasure to present this seminar, and I really thank Thomas for the initiative to organize this activity. So I will share my screen now to start my presentation.
Perfect.
Okay. Okay, so again, good morning from Costa Rica, good afternoon in Europe. I will cover today the topic of production and quality control of antivenoms, and I will try to present an integrated view of this topic. I work at the Instituto Clodomiro Picado, which is a research and production facility of the School of Microbiology of the University of Costa Rica. First, I will present an overview of how relevant this problem of snakebite envenoming is on a global basis. And if you see the figures in this map, it is estimated that about 2.5 million envenomings occur per year, and resulting in more than 100,000 deaths, and in about 300,000 people left with permanent sequela, either physical or psychological. So it's a big problem, and probably these numbers are much higher than the official numbers indicate. So the areas mostly affected are Asia, in particular India, Sub-Saharan Africa, and Latin America. Snake bites, which is an interaction between humans and snakes in particular contexts, occur in a specific ecological and socio-cultural contexts, and mostly affect, like shown in this picture, people living in impoverished rural communities. Therefore, snakebite envenoming has been considered a disease of poverty. And snake bites also affect livestock. So there is a complex interaction between humans, snakes, domestic animals, in a specific agroecosystem. So this integrated agroecological perspective is better captured by the concept of One Health, which integrates human medicine, veterinary medicine, and ecosystem variables, in particular health issues. So this idea of One Health is being promoted to better understand the way snake bites occur. There are many species of snake bites of snakes in the world, and here I show some of the most important in terms of the number of accidents and severity of the accidents. Here you see the Soskel viper, the puff adder, and the cobras in Africa, cobras also in Asia, the kraits and the Russell's viper in Asia, the lance head vipers in Latin America, and the taipan in Australia and Papua New Guinea. So this species and many others have evolved a complex system to synthesize venoms in a specialized gland and to deliver these venoms through an injection system, which plays a key role in the adaptation of these snakes to the environment for predation, for defense, and therefore when they bite humans or domestic animals, they mostly defensively, they inject these potent toxic secretions, causing a wide variety of clinical manifestations, which depend largely on the composition of the venom. So there is a great variation in venom composition, and this has been studied in the last decades in depth with the powerful tubes of mass spectrometry, which allow the proteomic analysis of venoms. And this has shown a wide variety of composition of the venoms from different protein families. And this variety of composition explains the spectrum, the wide spectrum of clinical manifestations induced by different types of toxins and venoms. Here I show just 4 examples. Here you see a typical neuromuscular paralysis inflicted by neurotoxins, mostly from elapid snake venoms, but also snake venoms, particularly viperid ones, but also some elapid ones like the spitting cobras, inflict local tissue damage, which results in cutaneous necrosis, muscle necrosis, necrosis of soft tissues, and the development of this type of blisters. And this local tissue damage is quite important because it develops very rapidly after the bite and usually results in permanent sequela. And also other type of systemic manifestations such as systemic bleeding related to the action of toxins on the blood vessels and on the coagulation system, which may cause a number of complications, including cardiovascular shock. So there is a wide variety, and this makes the topic of snakebite envenoming very complicated from the clinical standpoint and the therapeutic standpoint. The central point in the treatment of snakebite envenoming is the administration of antivenoms. And antivenoms were developed in the 19th century, in the year 1894, by Albert Calmette, who was appointed at Institut Pasteur and was working in what is today Vietnam. And he developed an antivenom against the cobra, the Naja, which was the first antivenom to be used clinically. Other researchers in France at that time, Physalis and Bertrand, also developed this idea that if you inject venoms into animals, they develop an immune response based on antibodies, which can be used therapeutically. In our region in Latin America, Vital Brasil Mineiro da Campanha started with Instituto Butantan in 1901 with the local production of antivenoms, particularly against South American snakes such as Bothrops yararaca and other snakes. And then in our own country, Costa Rica, Clodomiro Picado Twight studied the problem of snakebite envenoming, and he realized that the venoms from Costa Rica were similar to the venoms in Brazil, and therefore he made a cooperation with Instituto Butantan, through which antivenoms were provided by Brazil to Costa Rica. So even though he didn't start the production of antivenom in Costa Rica, he initiated the use of antivenom in our country. In Costa Rica, antivenom production started in the year 1967, and especially with the foundation of Instituto Clodomiro Picado in the year 1970. And then many other countries started the production of antivenom in different parts of the world. So basically, antivenoms are preparations made of immunoglobulins, either whole immunoglobulins, complete immunoglobulins, or their fragments, FAB2 or FAB fragments, which are purified from the plasma of animals that have been immunized with the snake venoms. The animal used by the vast majority of manufacturers is the horse, because it's highly convenient in many ways, but also some manufacturers use a sheep, particularly manufactured in the UK. The antivenom being used in the United States is made from sheep plasma. And also in locations such as La Paz in Bolivia, the local manufacturer uses donkeys and sometimes llamas. Also, some people have speculated, although this has not evolved into commercial manufacture, that camels could be used in some regions of Northern Africa and the Middle East. And experimentally, many research groups have produced antivenoms from the egg yolk of hens immunized with animals, but this also, as far as I know, has not evolved to commercial production. So the idea basically is to inject, and this idea started with Albert Calmet, like I said, in the 19th century, to inject venoms into an animal, allowing the immune system of the animal to manufacture antibodies against these venoms. So how these antibodies act, we will see in a while. I would like to mention that antivenoms have been introduced in the list, in the official list of the essential medicines of the World Health Organization, as you see in this document. So it's a drug, it's a medicine which is enlisted in this essential medicine list by the WHO. So briefly here, I would like to reflect on how antibodies in antivenoms or antibody fragments neutralize the toxins. So basically the action of these antibodies when they are administered in a patient suffering a snake bite envenoming is that these antibodies bind these toxins and neutralize or abrogate their toxicity and they promote their elimination from the body. So here I would explain that antibodies may act in different ways. These toxins that you see here is a metalloproteinase from the venom of Botrop asper from Costa Rica, which inflicts hemorrhage and severe tissue damage. So these metalloproteinases have an active catalytic site here, which includes a zinc ion, which is this orange sphere in the catalytic site. So an antibody can bind to the active site or the toxic site, so to speak, and therefore neutralize the activity of the toxin. But also an antibody may bind to an epitope located not exactly in the catalytic or active site, but in a different molecular region, and then induces inhibition through steric hindrance. So the antibody is large enough to block also the catalytic site, even if the epitope is not there. Another theoretical possibility is that antibodies may bind to sites distant from the catalytic site, but by binding to these sites, they induce a conformational change, an allosteric change in the toxin molecule, therefore blocking in this way their activity. And also, when they are divalent, such as IgG or FAB2, antibodies or antibody fragments may form aggregates, immune complexes, which block the action of the toxins and promote their elimination. So there are different ways through which antibodies can bind and inhibit particular toxins in a venom. The World Health Organization has issued an official guideline for the production and control and regulation of snake antivenom immunoglobulins. This process started in the year 2007, and both Professor David Warrell, myself, and other colleagues were involved in the preparation of the first version of these guidelines, which have been improved over the years. In the year 2017, there was a new edition of these guidelines, and a new one is being prepared. It is a very detailed document explaining all the aspects related to antivenom manufacture. So this is a guideline that should be used by manufacturers and people interested in production and control and regulation of antivenoms all over the world. In Latin America, from Instituto Clodomiro Picao, it has been prepared a Spanish version of these guidelines, which are openly available. So this is a very important document which presents many details related to antivenom production, and I will cover some of these details in the rest of my talk. Antivenoms are manufactured in many different countries in the world by a universe of laboratories, and some of them are public, some of them are private, some of them are large, some of them are small, some of them have very good infrastructure and technological platforms, some of them have weak and small technological platforms. So it's a highly heterogeneous universe or landscape of manufacturers. And you see there are some regions in the world, particularly sub-Saharan Africa, where there is a clear absence of manufacturers. And even though, therefore, there are many manufacturers in the world, there is a problem of availability and accessibility of antivenoms, which is being addressed by the World Health Organization and by many stakeholders in the world. So there is a need to improve the quality and the volume of antivenoms in the world right now. You can see that in Latin America, we have a good number of manufacturers, mostly in the public domain, which comprise an interesting collective of laboratories, which I will mention later on today. So the World Health Organization, as part of the global efforts to confront this disease, this neglected tropical disease, issued in the year 2019 a global strategy for the first time in history, a global strategy for the control and prevention of snakebite envenoming. And this strategy is based on four main pillars, which are the empowerment and engagement of communities, the strengthening of the public health system, the increase in partnerships, coordinations, and resources, and very important for our topic today, to ensure safe and effective treatments based mostly on antivenoms and some novel possibilities. And it is expected that as this strategy unfolds in the world, we will witness a reduction in the number of deaths and disabilities due to snakebite. And it is expected that by the year 2030, there will be a 50% reduction in mortality and sequela due to snakebite envenoming. So one of these pillars is to ensure safe and effective treatment, and then therefore to improve the antivenom quality and availability. Antivenoms in general are classified as either monospecific or polyspecific, depending on how many venoms you use in the immunization of animals. So for example, in Costa Rica, we produce a monospecific anticoral antivenom, which is used for the treatment of coral snake bite envenomings by species of the genus Micrurus in Central America. This is a monospecific antivenom because basically we use the venom of Micrurus nigrosynthus to produce this antivenom. On the other hand, for the treatment of envenomings by pit vipers, snakes of the family Viperidae, we prepare in Costa Rica a polyvalent antivenom. And it is polyvalent because horses are immunized with a mixture of the three species that you see here in the upper part, which are Bothrops asper, the fer de lance or terciopelo, Crotalus, the Central American rattlesnake, and Lachesis stenophis, the bush mussel. So by injecting several venoms, we are producing a polyvalent antivenom. It is important to consider that monovalent antivenoms or monospecific antivenoms, such as this one, doesn't mean that they only neutralize the venom which is used in the immunization, because it may cross-react with other species. So the word monospecific refers to the venom used in the immunization, not to the venoms which are neutralized by the particular antivenom. So the first step in the manufacture of antivenoms is, of course, to have a good source of high-quality venom. And for this, you need to keep snakes in collection. So manufacturers either have their own serpentarium, like we do in Costa Rica. And here are two colleagues from our institute, Danilo Chacon and Aaron Gomez, obtaining venom, or milking is the word used, from a very large Bothrops asper terciopelo from Costa Rica. So in our institute, we keep a collection of snakes, but some manufacturers purchase these venoms from providers of venoms, private companies or laboratories, which offer venoms for manufacturers. So either way, the quality, the maintenance of the snake, the good practices in the maintenance of snakes, and the ethical principles in handling the snakes and keeping the snakes are very important, as indicated in the WHO guidelines. So a key issue in antivenom production, and this is very important, is which venoms do you choose to immunize for immunization to prepare antivenoms of good neutralization coverage and good cross-reactivity. And this, for many years, this was done on a rather empirical basis. because there was not much knowledge about the composition and properties of snake venom. But now we do have a large and robust body of evidence, biochemical, toxicological, immunological, to be able to select the best mixtures of venom for immunization. So you need to consider for that which are the medically relevant species. For example, when our institute, in collaboration with the University of Oxford and the Liverpool School of Tropical Medicine in the decade of 2000, started the production of antivenom for sub-Saharan Africa, it was very relevant to select which venoms were medically important in Nigeria in particular. And in that case, the selection was based on X ocellatus, Vitis arietans, and Naja nigriculis, because the information obtained from clinical studies indicated that these were the three most important species. So information from the medical landscape is very important for the selection of species. Then the clinical manifestations of these envenomings allow sometimes the identification of the type of snake in the bite by what has been called a syndromic approach. So therefore, in many cases, it is not necessary to know exactly which species inflict the case, but the clinical syndrome allows the classification into several syndromic categories, which allow the selection of the venom. So the clinical information is very important, both for the selection of the venoms for immunization and for the treatment. Then another point which is emphasized in the WHO guidelines is that we need to have representative pools of venoms. And this is important because venoms have a great variation between species and within the species. So venoms from snakes located in different geographical regions, even from the same species, may vary, and in fact do vary. And also venoms vary according to the age of the snake. So there is a big ontogenetic change in the composition of venoms from several species, and these need to be considered. So pools representative of the variation in venom composition are important to have a good representation of the venoms of a particular snake species. And this demands biochemical and proteomic and this type of studies to select these pools. And then it is very important to have a quality analysis of the venoms, certification of the venoms, particularly if these are purchased from commercial providers, and then a concept that is present in the guidelines of the WHO, which is the traceability of the venoms. So it is very important to know which particular batch of venom is obtained from which snake, which was collected in which location, in which circumstances. So this traceability is absolutely essential for good manufacturing practices in antivenom production. So it's not just a matter, as you can see, of obtaining venom from snakes. They have to be, there has to be a very well prepared, standardized ways for venom collection, because everything starts with the quality of venom in antivenom manufacture. For example, I will give you 3 examples in Latin America, how venom mixtures are selected. As I mentioned, in the case of Central America, for pit viper, antivenom against pit viper envenomings include a mixture of Bothrops asper, Crotalus, and Lachesis stenophilus. But for example, in the case of Bothrops asper, we have a population of this species in the Caribbean side of the country and a different population in the Pacific side. They belong to the same species, but they have difference in venom composition. So in our mixture, we have a pool of venoms from both the Caribbean and the Pacific sides, and therefore, in that way, we cover this venom variation. In Brazil, for example, in 4 public institutes where they manufacture antivenoms, they select a combination for the bothropic antivenoms. They select a mixture of Bothrops jararaca, Jararacuzu, Alternatus, Mugini, and Newidi. And with this combination, they ensure cross-reactivity and neutralization of many other Bothrops species and species from other venoms from other species. And then, on the other hand, they prepare an antivenom against the rattlesnake from South America, which inflicts a very different clinical pattern of envenomations, characterized particularly by neurotoxicity and systemic myotoxicity and coagulopathy. So for that, they use the venom of Crotalus durissus terrificus and another subspecies, Crotalus durissus cascabella. and they manufacture this crotalic antivenom. So depending on the clinical manifestation, depending on the type of snakes present in a given region, you have to select different venom combinations. But as I said, we do have now enough information on venom composition and toxicity to make these selections based on knowledge and not just on empirical basis as it was in the past. I will illustrate this complexity of the venoms and the impact in antivenom design in the case of India. In India, there are several manufacturers which produce a huge volume of antivenom, and basically they use the venoms of what they call the big four snakes in India, which are the Lapids naya naya and Bungarus caeruleus, and the Viperids, the Boya russellai, and Echis carinatus. However, the source of venoms come from a very particular region in India, and therefore there is a need to include venoms from other regions which may bring the representation of this variety of venom composition. So this is being discussed actively in the antivenom field. And in addition to considering the variation of venom, for example, the venom of the Boya russellai varies depending on the geographical region where the venom is collected. In addition to that, clinical evidence indicate that other species are also relevant from the snake bite and venom perspective in India, such as the hawk-nosed viper Ibnale ibnale, which inflicts a large number of cases in India, Sri Lanka, and so on. So there is a need to consider the addition of the venom of Ibnale ibnale for the antivenom manufacture in India. So this may harness all the knowledge that is being obtained in venom composition to improve the mixture of venoms used in antivenom manufacture in India and elsewhere in the world. Here I show a map with from a work of Davinia Pla and colleagues from the group of Juan Jose Calvete, showing what is shown here is the different isoforms of phospholipases in different samples of venoms of the Boya russellai, of Rossus viper, in different parts of Asia. Basically, what I want to show is that there is a great variation in the types of phospholipases that these snakes present. Therefore, illustrating this geographical variation in venom composition, which should be taken into consideration for the selection of venoms for antivenom manufacture. So the preparation of good quality venoms is, as I mentioned, very important for antivenom production, and this is stressed in the WHO guidelines. It is important to avoid proteolytic degradation of venoms. I mean, snake venoms, particularly viperid venoms, contain a high content of proteinases, and if the venom, once obtained, is allowed to stay at room temperature for a given time, there could be proteolytic degradation of venom. So this should be taken care of. It is recommended that once venom are obtained after milking, they should be rapidly frozen, and then stored frozen, and eventually they should be freeze dried through lyophilization. Some laboratories, but this is not the case in most of them, use desiccation of venoms, but it is preferred to use a freeze drying of venoms. And this will make venoms very stable and can be stored in the freezer for very long periods of time. So the way venoms are collected and handled and prepared is key for a good quality antivenom. And then, as I mentioned before, traceability is essential. I mean, to know the venom pool from which particular specimens come, where these specimens were obtained or collected, and what has happened to these snakes in the period of captivity, and so on. So laboratories that provide venoms or antivenom manufacturers that have serpentarium should ensure the quality of venoms through appropriate quality control. And this quality control involves at least the regular analysis of residual humidity in freeze-dried antivenoms, because freeze-dried products tend to accumulate humidity, And then chemically, the quality of the venoms and the identity of the venoms can be regularly checked also through, for example, SDA's phage electrophoresis, polyacrylamide electrophoresis, or chromatography through reverse phase high liquid performance chromatography, high performance liquid chromatography, HPLC. So you have a particular pattern of HPLC and of electrophoresis in particular venoms, and this is a way to control the identity and the quality of the venoms. And also, it is important to control the toxicity of the venoms, either by doing the median lethal dose, the LD50, or functional properties such as in vitro coagulant activity, proteolytic activity, phospholipase activity, and so on. So that's another way to ensure that venoms are active and of high quality. So this is very important. And for laboratories, manufacturing laboratories that purchase the venom from commercial providers, it is critical to request this particular type of quality control analysis. So once you have selected the venoms for immunization and you have collected the venoms in appropriate ways, the next step is, of course, the immunization of animals. So first of all, animals, particularly horses, which are used by most manufacturers, need to be obtained, maintained and taken care of in very standardized protocols, which are also indicated in the WHO guidelines. And therefore, the selection and veterinary care of animals is essential. Vaccination, deparasitation, control of infectious diseases, control of hematological and clinical chemistry, and clinical parameters of the horses to ensure that horses are in good condition and good health for antivenom manufacture. So this is absolutely relevant for the quality of antivenoms. And there should be protocols with traceability also regarding the quality and the health status of horses. So then laboratories should prepare immunization schemes. So how horses or animals are immunized, what is the dose of venom, the amount of venom injected. It is injected in a single site of different anatomical sites. And then venoms, and then at what time intervals the venoms should be injected. And different laboratories have different schedules for immunizations. But also the understanding of horse immunology and the immune response is quite relevant for an appropriate design of immunization schemes. One relevant point is that immunization needs to use adjuvants. Adjuvants are substances of different sorts. It could be emulsions or salts or different molecules of different compositions, which enhance the immune response of animals. So always the immunization of animals for antivenom production should include adjuvants, such as fluorine complete adjuvants, aluminum hydroxide, or novel adjuvants being developed. And this is a field of research that needs to be strengthened, because with better adjuvants, we will get better antibody responses in horses. And then there should be a permanent monitoring of the immune response and the animal health. So the participation of professionals in veterinary medicine to take care of these horses and follow up their clinical developments and the immune response is very important. This figure here in the bottom is from the WHO guidelines and shows different regions where venom can be injected, which are close to places where lymph nodes are located, therefore stimulating a better immune response in theory. There is a group in Thailand by Dr. Kavi Ranatabangun, which has proposed the use of injection not in a single site in the horse, which is usually a subcutaneous injection, but in different sites, like 10 different sites. This is called low volume multi-site protocol of injection, and it has been demonstrated that it induces a higher antibody response. So this is a very important part of the whole antivenom production process. It is relevant to keep in mind that different toxins in the venom have different immunogenicity. And this is sometimes gets to be a big problem for antivenom manufacturing. For example, here I show two different types of metalloproteinases, which are key components in snake venoms. Here you have what is called a P3 metalloproteinase. It's a large enzyme, whose molecular weight ranges between 50 and 70 kilodaltons. This is a very good immunogen because in immunology, it is a rule of thumb that the larger the antigen, the better the immune response. And this rule applies in the case of antivenoms. So responses to P3 metalloproteinases, such as hemorrhagic toxins or procoagulant toxins, is usually very good and very high. In contrast, when we work with P1 metalloproteinases, whose molecular mass is about 220 kilodaltons, the immune response is lower as compared to the P3 ones because the lower molecular mass. And then the main problem in antivenom production right now is to deal with these small type of toxins, which have 6 kilodaltons mass, which are called three-finger toxins because they have 3 loops in the structure. And these toxins are neurotoxins and cytotoxins, which play very important roles in toxicity, particularly in elapid venoms. So, for example, what I call alpha neurotoxins, they bind strongly to the cholinergic receptor and induce neuromuscular blockade and paralysis, respiratory paralysis and death. And some of the cobras, particularly the spitting cobras, have a three-finger toxin type of toxin called cytotoxins, which induce cutaneous tissue necrosis. And these toxins, by being so small, it is very difficult to raise a good antibody response against these toxins. So many groups from different parts of the world are working, trying to see how to improve the immune response against this particular type of toxins. So the immunogenicity of toxins varies, and this has an impact in antivenom production. Once the horses or the immunized animals have developed a strong and robust antibody response, which can be followed by immunoassays such as ELISA, for instance, then it is time to obtain blood from these horses. Again, this has to be done with care and taking into consideration the health status of the animal. So a volume of blood is obtained from bleeding, usually from the jugular vein, as you see here, and is collected in plastic bags, sometimes in glass bottles, but usually in plastic bags that contain an anticoagulant, citrate anticoagulant. So every laboratory establishes the schedule for bleeding of horses. It could be 3 consecutive days, it should be 1 day only, or separate days in time. And this needs to be, of course, validated and reproduced in the manufacturing laboratories. So blood is collected, and then plasma is separated from the red cell and cell component in the blood. So usually in the horses, red blood cells spontaneously sediment to the bottom of the bag, and there is no need for centrifugation, which is a good thing because it makes the separation of plasma easier than if you would have to centrifuge the blood. So usually the red blood cells are resuspended in saline solution and are reinfused to the horse. This corresponds to a plasmapheresis, which can be done manually. I mean, allowing the blood to sediment, then prepare the suspensions of red blood cells and reinfusing this into the animal. Or it could be done, and some laboratories do that, by automatic plasmapheresis. This is plasmapheresis. This is separation of red blood cells and plasma through a centrifuge, which enables the immediate replacement of red blood cells to the horse and increases the yield of plasma. And several laboratories are testing this new or this alternative modality of collecting blood. In our institute in Costa Rica, we collect blood in this way. We allow the cells to sediment, and then we reinfuse the red blood cell suspension the next day. So once we have the plasma in these bags, there should be a quality control of this plasma before it goes to the fractionation laboratory. And again, the concept of traceability is very important. I mean, to know from which horse that particular pool of plasma was obtained, which venoms were used in the immunization of horses, which health issues these particular horses had, and so on. Plasma usually stored between 2 and 8 degrees Celsius, and then big batches of plasma are prepared for fractionation. And again, here, as well as in the rest of the manufacturing process, the follow of good manufacturing practices is essential and is a mandate in the WHO guideline for antivenom production. And this involves many different issues regarding to the preparation of the equipment, the use of clean rooms, the preparation of water. Every aspect of antivenom manufacture should be done following good manufacturing practices. So the next step is the fractionation of the plasma, because plasma is a soup, is a mixture of many different proteins. Among the different proteins, you have the antibodies, which are the proteins that we want for the antivenom, but also you have large quantities of albumin, other globulins, fibrinogen, and many different proteins. So antivenoms in many laboratories are made of whole IgG molecules, whole immunoglobulin molecules, which is represented by this central structure here. But many manufacturers, in fact, most manufacturers, use a protocol based on the application of the proteolytic enzyme pepsin, which works at acidic pH. So you need to drop the pH to about 3 or 3 something, and add pepsin. And basically what pepsin does is to cleave the antibody in this site, releasing this part, which is the called FC fragment, and then leaving only this part, which is the part that combines with the antigen, which have the FAB fragments. And this is called FAB2 because it has 2 FAB fragments. So most antivenoms in the world are made of FAB2 fragments. Some, like in our case in Costa Rica, are made of all IgG molecules. And one manufacturer, the one that distributes antivenom in the United States, treats the plasma not with pepsin, but with another proteolytic enzyme called papain, which works at neutral pH, and cleaves the molecule here above these disulfide bridges, therefore generating 2 monovalent fragments called FAB fragments. So there are 3 types of antivenoms in terms of the nature of the active principle, either FAB, whole IgG, or FAB2. The WHO guideline for antivenom production accepts the 3 formats. However, it has been indicated by a number of studies that FAB products, the monovalent products, have a pharmacokinetic profile which impacts in the half-life of these products. So the concentration of antibodies or antibody fragments, FAB fragments, after intravenous administration drops very rapidly, and this demands a protocol of administration based on repeated infusions of the antibody fragment preparation to keep a high concentration of these fragments in the blood. This pharmacokinetic problem, so to speak, doesn't occur with IgG and FAB2 antivenoms, which remain in the circulation for more prolonged periods of time. But the 3 types of fragments are accepted in the WHO guidelines. I will briefly review here, without getting into much detail, how is this fractionation or purification of antibodies or antibody fragments, how it is done. In our case in Costa Rica and other laboratories in the world that use caprylic acid precipitation, the process is basically the following. Plasma is mixed with caprylic acid at a concentration of 5% and a relatively acid pH. And what caprylic acid does is to precipitate all the proteins in the plasma except the immunoglobulins. So after filtration, you collect a filtrate or supernatant, if you centrifuge, which is rich in immunoglobulins, and then the precipitate contains all the rest of plasma proteins. So it's a very convenient protocol for antibody production. Then the preparation, the pH of the preparation is adjusted. It is submitted to a process of diafiltration through a membrane that allows the separation of small molecular fragments and caprylic acid and so on. And then the product is concentrated to the protein concentration that would achieve a neutralizing efficacy or ability of the antivenom. So we have an IgG solution, which is formulated and then is filtered through a sterile filtration. That means through a membrane which has a pore of 0.22 micrometers, and this sterilizes the product. So you have the bulk product, which is then bottled to have the final product in 10 milliliters vial. So this is a very simple protocol which yields a preparation of high purity. In contrast, the laboratories, which are the majority in the world, which produce antivenoms based on FAB2 fragments by pepsin digestion, what they do is initially they add pepsin and acidify the preparation to reach acidic pH of plasma. And this cleaves antibodies and then also degrades many contaminant proteins in the plasma. Then there is a first precipitation with ammonium sulfate, a process called salting out at 12%. And the antibodies fragments are in the supernatant. So this is filtered or centrifuged. And then the supernatant is submitted to a process of thermocoagulation. Temperature is increased and filtration again. And this also precipitates non-IgG proteins. And this is also centrifuge or filter. And then this second supernatant is submitted to a second ammonium sulfate precipitation at 23%, which in this case precipitates the FAB2 fragments. So after filtration, you have a paste, a precipitate of FAB2, which is then dissolved in a saline solution and submitted to diafiltration to eliminate ammonium sulfate and other small molecules, formulation, filtration, and then bottling. So these are two different protocols, and they are designed to obtain either whole IgG or FAB2 fragments. So these are 4 pictures from our production laboratory in the Institute to show you. Here is Michael Cerdas preparing the diafiltration process through this external control of the clean room. And this is Eduardo Segura after the bottling of the antivenom, selecting the vials and so on. This is Christian also checking for the composition of the quality of the bottles and the different aspects of the manufacturing process. So this is a very complex process. Again, it demands good manufacturing practices, the design of the clean rooms, and the protocols in many aspects, which are clearly described in the WHO guidelines for antivenom production. Then antivenoms are distributed or formulated in 2 different formats, either liquid or freeze-dried. So you see here another colleague from our institute introducing a batch of antivenom, liquid antivenom, into a freeze dryer for the process of freeze drying. So in many cases, antivenoms are bottled as liquid preparations, like this polyvalent antivenom from Costa Rica. And in our case, several countries use liquid antivenoms, like Costa Rica, Panama. Even an antivenom that we produce for sub-Saharan Africa is currently manufactured in liquid form. So this is a ready-to-use product, which is just to be obtained from the vial. But in many countries require freeze-dried antivenoms because they are more stable and can be stored, as we will see, at room temperature. So for this, it is necessary to develop this process of freeze drying, which basically consists in the freezing of the product to -40 degrees Celsius, and then a process of sublimation, whereby the water is eliminated from the product, and then a process of secondary drying and eventually the product is obtained in solid form with very low amount of water. So there are differences and contrasts between liquid and freeze-dried antivenoms. For instance, there are differences in stability. Liquid antivenoms in general require a cold chain. That means that during storage and distribution, the product has to be kept between 2 and 8 degrees Celsius. And this is sometimes gets to be a heavy demand for the public health systems. That doesn't mean that if antivenom is out of this temperature for a few minutes, it would get ruined. But formally, it is required that antivenoms have to be stored between 2 and 8 degrees Celsius if they are liquid. It has been observed by different studies that liquid formulations are more stable than previously thought. And therefore, there is a need to further study the stability of liquid formulations and to innovate in the formulation, in the composition of liquid antivenoms to increase their stability, to increase the shelf life. Officially, the shelf life of liquid antivenoms is 3 years, whereas freeze-dried antivenom is 5 years. However, it has been observed in a number of studies that this shelf life is probably more prolonged for these two products, but this has to be, every manufacturer needs to study the stability, the shelf life of their particular products and validate this through stability studies. That's the way they are called. In terms of convenience and easy to use, liquid antivenoms are more convenient, and this is why many countries prefer and users prefer liquid antivenoms. And then there is the issue of cost, because freeze-dried antivenoms are usually more expensive than liquid antivenoms. But stability is very important, and for some regions of the world, freeze-dried antivenoms are strictly necessary. Then the process of freeze-drying should be done in a very careful way in order to ensure that aggregates are not formed and the safety is good enough. A clinical trial is right now underway in Sri Lanka to compare liquid and freeze-dried antivenoms produced in Costa Rica and venoms in Sri Lanka. Some laboratories use additional steps, such as ion exchange chromatography. There is a need to introduce steps for viral removal or inactivation. And in some manufacturers that produce FAB products, caprylic acid is also used to eliminate lipoproteins. And as I mentioned, the formulation of the products is being could be improved through the addition of osmolytes. Then, once the antivenom is manufactured, the quality control is very important. Here is our colleague, Gabriela Solano from Instituto Clodomiro Picado, the quality control laboratory, doing an assay for quality control of a batch of antivenom. And it's important to remark that quality control is important not only at the end of the process, but also along the process. So there is in-process quality control and quality control for end product. I will not get into much detail because we don't have much time, but I like to mention that the quality control tests involve biological tests, chemical tests, and additional analysis. And in particular, it's important to check for sterility, for the presence of endotoxins or pyrogens, and of course, to ensure that the neutralizing potency of the antivenom is good enough. So an antivenom should have a neutralizing potency. For example, our Central American antivenom, 1 milliliter of antivenom should neutralize 3 milligrams of Botrop Sasper venoms in the lethality assay. The same value holds for X ocellatus venom for our antivenom for Africa. Every antivenom has a specific potency that has to be reached for the batch of antivenom to be released. And there are, again, as I mentioned, chemical tests and additional tests. So I show you here just briefly an example of a test that is recommended in the WHO guidelines to detect the presence of molecular aggregates. When an antivenom has molecular aggregates of immunoglobulins, this antivenom is more prone to induce adverse reactions. So here we compare 2 IgG antivenoms, one made of with ammonium sulfate precipitation and one with caprylic acid. These were compared in a clinical trial in Colombia coordinated by Dr. Rafael Otero Patiño. And you see that in the case of ammonium sulfate derived product, this is a gel filtration. It's a chromatographic separation of gel filtration, which separates molecules on the basis of molecular mass. And you see that the ammonium sulfate made antivenom has protein aggregates here in the high molecular mass components, which are almost absent in the case of a caprylic acid antivenom, and also has some albumin, which is not present in caprylic acid manufactured antivenom. And accordingly, the incidence of adverse reaction of these two products when tested in a clinical trial was higher in the case of this antivenom as compared to this one. So this is an example of a quality control test that is recommended by the WHO. Then antivenoms have to be evaluated at the preclinical level for their efficacy. And this has been done for a number of antivenoms, but unfortunately, there are many antivenoms in the world which have not been tested for their preclinical efficacy. And preclinical efficacy means to test whether antivenoms neutralize relevant toxic activities of venoms in experimental models, usually in mice. So for example, for pit viper venoms in Latin America, it is necessary to show not only that the antivenom neutralizes lethality, which is the gold standard proof of potency, but also that antivenoms are able to neutralize hemorrhagic activity, procoagulant activity, defibrinogenating activity, and myotoxic activity. So these type of studies have been done in Latin America by our groups in collaboration with other groups, and there are protocols for these tests, like in this manual developed in Costa Rica and used in Latin America, and also the WHO guidelines has a detailed protocol of preclinical tests. So I'm showing here two studies, one comparing antivenoms from Brazil and Costa Rica against venoms from Latin America, the antivenom from Costa Rica and Peru against Peruvian venoms. And this is probably the largest preclinical antivenom study done by comparing different Bothrops venoms in Latin America against different antivenoms in our region. And it was found, and this is interesting and encouraging, that most antivenoms have a good cross-reactivity against many Bothrops venom in the region, indicated that antivenoms from particular countries can be used in other countries in this type of envenomings. And then, ideally, antivenoms should be also tested in well-designed clinical trials for safety and efficacy. So the final proof of efficacy and safety of antivenom should be done in well-designed clinical trials, ideally randomized, controlled, double-blind clinical trials, such as this one, which is probably the largest clinical trial ever done with antivenoms, comparing two antivenoms in envenomings by Equus ocellatus, the carpet viper, in Nigeria, and published in the year 2010. So the WHO is encouraging health authorities, researchers, and manufacturers to validate clinically the safety and efficacy of antivenoms. Another point which is developed in the guidelines is the need to strengthen the national regulatory agencies, because right now in many countries, antivenoms are introduced without a proper validation of their efficacy and safety. And this is the responsibility mostly of the national regulatory agencies that belong to the ministries of health. So sometimes the name of a product may bring confusion. For example, saying polyvalent antivenom, a person may think that polyvalent antivenom would work for any type of venom, and actually has a specificity for particular venoms that have been used in the immunization. So this brings confusion. And then countries need to ensure, as I was saying, that antivenoms that are imported and indeed effective against venoms from the country where they are going to be used. So for this, the national regulatory agency need to be strengthened through regional workshops, training activities, and cooperation. This is just to show that in Latin America, we have been able to build a network, which is called RELAPA, the Network of Public Antivenom Manufacturing Laboratories of Latin America, with many laboratories related to production or control of antivenoms in different countries. And we meet regularly under the coordination of the Pan American Health Organization, the office PANAFTOSA of PAHO, regularly to have workshops technical exchanges, mutual visits, research projects. So under a frame of cooperation and generosity and solidarity, we reinforce the regional capacity to produce and control and distribute antivenoms. Also, getting to the end of my talk, it is relevant to mention that snakebite envenomy is important in veterinary medicine. And even though this problem has not been documented in many parts of the world, recent studies done in Costa Rica, Nepal, and Cameroon have shown how important this is for livestock and how big the impact of this problem has for the economy and social aspects of the communities that rely on this livestock for their living. So very few countries manufacture antivenoms for veterinary use. And in our case, in Costa Rica, we manufacture this veterinary polyvalent antivenom, which is distributed in our country, and now it's going to be distributed in other countries as well, for the treatment of animals bitten by suffering snakebite envenoms. Now, beyond antivenom production and quality controls, there are other aspects that need to be considered in the field of antivenoms. So, for example, do health authorities purchase the volumes of antivenoms required? And what is the volume of antivenom that a particular country obtains for distribution? Sometimes it is insufficient. Is antivenom distribution to health posts, especially to rural health posts, effective? I mean, do antivenoms reach rural health posts where they are mostly needed? Are there national guidelines for diagnosis and treatment of envenoms or regional guidelines? Do health professionals know the basic aspects of the management of snakebite envenoms? Because you may have a good antivenom, the country may purchase a good volume of antivenom, it may distribute it to the rural health facilities where snake bites occur, but if the health staff, physicians and nurses and other health staff working at these health facilities do not know the basic aspects of antivenom selection, administration, treatment of adverse reactions and complications, if they do not know these aspects, then the treatment will be ineffective or even harmful. And then, are there pharmacovigilance programs to follow up the performance of antivenoms in the field? So these are questions related to antivenoms, not necessarily to antivenom production, but for distribution and use of antivenom from a public health perspective. So for example, the question, do antivenoms reach regions of highest need? So in many cases, there is poor epidemiological record in some countries which do not allow the health authorities to deploy antivenoms where they should be deployed. So a sound epidemiological information is necessary for the distribution of antivenoms. Sometimes ministries of health have low budget and slow processes of acquisition. Sometimes antivenom is concentrated in city hospitals and not in rural settings. In the case of liquid antivenoms, in many countries, there are problems with the cold chain. So the cold chain cannot be assured during storage and transportation of the antivenom. There is deficient provision in many cases of health services in remote rural settings. So people need to travel long distances to get to the nearest health facility where antivenoms are available. And therefore, the question is how to ensure to have not only a good availability of antivenoms, but also a good accessibility of antivenoms, so that a person bitten by a snake in a remote rural location can reach a health facility rapidly and can receive an effective and safe antivenom for treatment. So this is an open question that involves, of course, many aspects of public health and health politics in many parts of the world. And this is being also considered within the frame of the WHO strategy for antivenom manufacture, distribution, and for the treatment of snakebite envenom. So in Costa Rica, for example, a study done by Eric Hanson on the distribution of antivenom and health services. Here you see basically the incidence of snakebite envenoming at the district levels. So you see regions in the Pacific side of the country, the Caribbean side, and the northern part of the country with highest incidence, basically due to envenomings by Bothrops asper. And here you see the distribution of health facilities, basically hospitals and clinics in our country. And Hanson was able to detect several regions in Costa Rica where the transportation of a person to the nearest hospital takes more than 3 hours. So these are regions where it is important to deploy antivenoms to primary health facilities to ensure that people will receive the antivenom rapidly after the bite. So this type of geographic analysis of health facilities, distribution of snake bites, and location of antivenoms are very important for the policymakers all over the world. So here I stop, thanking Thomas and all of you for the invitation, and all of you for your presence and your attention. This is the entrance of Instituto Clodomiro Picado. You are all welcome to visit us when you are in Costa Rica. This is my email address in case somebody wants to send some questions or comments. And again, thank you very much. I look forward to hear your comments and questions to my presentation.
Thank you very much for taking the time to make the presentation. Very much appreciated. So, can you hear me?
Yes.
Okay, fantastic. So there are a few questions. I have received some personally, and then there's others in the chat. So for anyone that would like to ask a question, you can use the Q&A, but I will try to read some of the ones that are already in the chat section. So the first question from Cesar Renifo, if I'm pronouncing this correctly, is the WHO new guidelines including hens IgY antivenom manufacture?
No, they are not including that because there is no enough evidence, technological evidence that have scaled up the production of hen-derived antibodies. So this is not included. It's mentioned, but it is not included in the protocols.
Mm-hmm. There's another question that reads, what is the average time for the administration of a life-saving antivenom immunoglobulin injection? They say, I ask about the therapeutic window of administration.
Yes, that varies a lot depending on the severity of envenoming, the type of snake that inflicted the envenoming, and the patient itself. So, for example, there are cases of elapid venoms like mamba venoms in which the clinical manifestations develop very rapidly after the venom. So in that time, the therapeutic window, the window of time for administration of antivenom is relatively small. There are other cases in which the development of the symptoms and signs of envenoming take more time, and also depending on the severity of the case. For example, in a bite by Bothrops atrox or Bothrops asper, whereby the snake injects a large volume of venom, the development of local tissue damage is very rapid, and therefore, if the antivenom is delayed several hours, probably this person will have permanent tissue loss or permanent tissue damage in their body. So it also depends on the characteristics of the patient. Usually, envenomings in children are more severe and develop more rapidly than in adults. So it varies a lot. But the rule of thumb here is that the longer the time it takes to receive the antivenom, the poorer the prognosis of the case. And this has been shown in several regions of the world where this is almost a direct correlation between the time of administration and the complications of the bite. Mm-hmm.
So a question following that one is the lyophilized venom injected in the immunization host heated or chemically neutralized prior to the administration?
Mm-hmm. In general, antivenom is administered in its native form without any type of treatment, because it is feared that this type of treatments, especially heating, or for example, mixing it with the glutaraldehyde, may alter the epitopes of the toxins and may affect the immune response. So the way this has evolved is through the optimization, the immunization schedule by using lower amounts of venoms. So this is our case in Costa Rica, where the amount of venom injected has been reduced, and therefore, even though the venom is not detoxified, the health of the horses is not affected significantly in these cases. Mm-hmm. Some people use inhibitors, some people treat the venoms in some ways, but this is risky because the possibility of altering the antigenic characteristics of the venoms. So I think most manufacturers do not treat the venom before injection, except by adding adjuvants, which do not affect the quality of the venom.
Mhm. And following this question is another one. Some of the venom components may be valuable on an individual basis as natural potent pharmaceuticals. Is there an international database for such compounds?
No, yes, this is absolutely true. So we can see venoms as a source of a medical problem in terms of envenomings, but also as a potential source of new drug leads for pharmaceutical development. And indeed, there are several drugs in the market which have been developed from venom components. Perhaps one of the most typical examples is these antihypertensive drugs, enalapril and captopril, which were derived from bradykinin potentiating peptides found in the venoms of South American snakes. And basically they drop the blood pressure, and they are very effective antihypertensive drugs. So many groups are developing this line of research, trying to find new drug leads in many ways against several diseases. But there is, I don't know, I don't think there is like a website or an international list of these products. You can find them in the literature, and there have been some review articles which list all the products that have been developed from venom sources. Many groups are working in this particular area, and this is absolutely promising because of the high variety and versatility in action of venom components.
Thank you. Another question from Yanis Ariyanis, all the way from Cyprus. He says, great talk. Do we know which peptide or other compound of the venom binds to the Fab region?
To the, sorry?
To the Fab, F-A-B region.
Which venom components?
Yes. Which peptide or other compound of the venom binds to the Fab region?
Okay. Mm-hmm. Well, the first way to approach this is just to do with purified toxins to test whether antivenoms bind to particular toxins. And Professor Juan Calvete has developed, and his group, they have developed what is called antivenomics, which means that you put the antibody or the antibody fragment in a chromatographic column, and then you pass the venom, and then you detect which toxins are being bound by venom components, and you can identify these toxins. This has been a powerful tool to characterize towards which toxins antivenoms have antibodies. Now, if you, in a particular toxin, let's say that you demonstrate that an antivenom FAB or whole IgG has antibodies against a metalloproteinase, for example. If you want to know which are the epitopes in this particular toxin towards which antibodies bind, then this can be done by synthesizing the different sequences in this molecule by, for example, 13, 14 amino acid sequences, different. And then you test the antibodies against these particular sequences. In that way, you can find which sequences are recognized by the antibody. Of course, this refers to what is called linear epitopes, because this is a linear synthesis of a peptide. It doesn't recognize what is called conformational epitopes, which depend on the 3D structure of the epitopes. But that's the way people have studied where are the epitopes in particular toxins. And in fact, this recognition has also enabled the design of recombinant antigens, what is called multi-epitopes. So you select the epitopes, and then you made a construct, a recombinant construct having these epitopes, and then you can immunize with these epitopes. So this is something that I didn't mention. There is the possibility of, instead of using crude venoms for immunization, to use purified toxins, either isolated or synthesized in a recombinant fashion to immunize. And this is, for example, this is being done commercially already in the case of the venom of the spider Loxosceles, the brown recluse spider, which whose toxicity is based on a sphingomyelinase, and some groups are immunizing with recombinant sphingomyelinase. This can be done also for snake venoms, and this would result in benefits in terms of the standardization and the production of the antigens.
Thank you. Next question, Professor again, phylogenetics. Elias Capaz, he's asking, is there a next generation approach for the therapy of envenomings apart from antivenom production?
Mhm. Yeah, this is a good point. I didn't mention this because my talk was focused on current antivenom manufacturers, but thank you for asking this. Yes, in fact, in the WHO strategy for control and prevention of antivenom, it is mentioned and proposed that new lines for development, new type of therapeutics should be promoted parallel to animal-derived antivenom production. And the two main lines, I would say, are the use of recombinant antibodies, which could be of human origin or chimeric or other type. And several groups in the world are working in this area to identify the toxins and to produce recombinant antivenoms, monoclonal or oligoclonal mixtures of recombinant antibodies that would react against these toxins. So this is being developed, and there is a future for this line of recombinant antibodies or antivenoms. And the other line, which is yielding very interesting results, is the search for inhibitors, either natural or synthetic inhibitors that would block particular toxins. And I would like to mention particularly two types of inhibitors which are being used, which are metalloproteinase inhibitors and phospholipase inhibitors. These inhibitors have been developed by the pharmaceutical industry for other types of diseases, in which endogenous phospholipases or metalloproteinases play a role, such as cancer, arthritis, inflammatory diseases, cardiovascular diseases. So these inhibitors already went through phase 1, phase 2, and sometimes phase 3 clinical trials. These inhibitors are being repurposed, reintroduced to be used in case of snakebite envenoming, because a phospholipase inhibitor, for example, the case of varesclavir, can inhibit every phospholipase from different venoms, regardless of their antigenic characteristics, immunologic characteristics. So it's a very powerful, potentially powerful tool. And it has been shown that these PLA2 inhibitors preclinically are very effective against different types of venoms. And there is an ongoing clinical trial in the United States and India with varesclavir, the PL2 inhibitor. And something similar occurs with metalloproteinase inhibitors, in which chelating agents are being considered for inhibiting metalloproteinases of venom, or another type of inhibitors called peptidomimetic inhibitors. So probably in the future, ideally, we will have a combination of classical antivenoms, which always will be at least for years to come, the mainstay in the treatment of snakebite envenoming. So we should work in this area, improving the production, the quality, and the output of traditional animal-derived antivenoms, but also these lines of recombinant antibodies and synthetic inhibitors, if they develop properly, will bring novel possibilities to complement antivenoms in this therapeutic area.
Mm-hmm. And he responded saying, thank you. Are there any bioinformatics computational approaches so far when it comes to antivenom discovery?
Yes. Several groups have done, because with all the databases, with the sequences of toxins, several groups have introduced bioinformatic approaches to select the epitopes in the different types of toxins. So there is, in theory, the possibility of designing multi-epitope constructs based on bioinformatic analysis of sequences that would include the most important epitopes of different types of toxins. So it is feasible to have a construct that has epitopes for phospholipases, for three-finger toxins, for metalloproteinases, serine proteinases in a single molecule, and using this molecule for immunization. So this is being done. Several studies have been published, although, as far as I know, this has not translated yet to the technological, to the manufacturing realm. But this is a very clear possibility. So this shows how important is basic science for its applications for antivenom production. So the more we know about toxins, the more we know about the epitope composition, the more resources we will have to improve antivenoms.
Mm-hmm. Now I've gotten two questions that I think you have in part answered. The first one refers to the fact how far off would we be from producing monoclonal antibodies, the next generation and whatnot?
Yeah, well, like I mentioned, the technology is there. Several groups are working in this. Several projects are being supported to do this, and it is, I mean, it is absolutely feasible. One thing that makes this complicated is that snake venoms, as I mentioned, are very complex cocktails of different toxins. So depending on the venom and the predominant toxins, then you may need to have a cocktail of monoclonal antibodies. In the case of some venoms where a single toxin plays a key role in toxicity, it is easier to design one or few monoclonal antibodies that would block these toxins. But in the case of venoms that had more toxin, then you would have you will need to have a cocktail of oligoclonal antibodies. Of course, there is also the possibility of adding some monoclonal antibodies to animal-derived antivenoms and enrich the animal-derived antivenoms with monoclonal antibodies. That's another possibility. But all of this is open for research and development, and the possibilities are there, of course. I mean, the technologies are there already.
And another question has been partially answered. It reads, except for antivenom research, are the venoms also interesting from other perspectives, for example, against cancer, autoimmune disease, and so on?
Yes, absolutely. And as I mentioned, many groups are working, trying to isolate venoms in terms of looking for lead molecules for pharmaceutical development.
Another question is, are liquid or freeze-dried antivenoms of better quality?
Of better quality? No, in terms of biochemical composition and neutralizing potencies, it's the same. I mean, you have a batch of liquid antivenom, you freeze-dry the antivenom. If you do the freeze-drying in a proper way, as should be done, then these two antivenoms would have similar composition, similar neutralizing potency. The problem comes sometimes when the freeze drying is not done properly, because then you introduce molecular aggregates. Sometimes the potency may decrease and the quality of the product may decrease. But if freeze drying is done properly, then the two products would be similar in terms of neutralizing potency and biochemical characteristics.
So another question is from Eleni Ionidou. She's saying, thank you for your speech. It was very interesting. I would like to ask, how can we be sure that the volume of venom injected to the horses isn't harmful to them?
Well, this is done through a very careful observation and control, veterinary control of the horse's health. So for example, in our case, every injection of venom, like I said, the amount of venom has been reduced, is followed by a careful control of the health status of the horse, both in the laboratory and in the clinical aspect. So this should be carefully followed, and this allows this allows the health of the horse to be well kept and cared for. This is very important, and therefore the veterinary component in this whole picture of antivenom manufacture is key.
Really, yes, because it's not as widespread information as one would have thought, especially here. Another question is, what's the cost for the production of a bottle of antivenom in liquid form?
This is a very important question, and it varies enormously between different manufacturers and different contexts. So antivenom production laboratories keep a detailed control of all the costs involved in the production of the antivenom, so that the cost of a particular vial is determined, and therefore the price of the antivenom is defined on the basis of that cost. But this cost varies enormously depending on the laboratory the particular laboratory, and this may explain partially the huge differences in the prices of antivenoms in the world. This is another aspect in which the WHO is insisting that the more good quality products are in the market in a particular region, then the competition between manufacturers is introduced, and then probably the prices would go down as badly needed in public health systems of developing countries. So this issue of price and cost is very important, and WHO is thinking along this line.
This segues nicely with a question I got, which says, do you think the number of antivenom production labs is enough? Do they all have close ties to universities?
No, it's very heterogeneous. You have laboratories like our laboratory in Costa Rica, which is tied to the university. You have other laboratories like Instituto Butantan, for example, which is tied to the Secretary of Health of the state of Sao Paulo. You have private laboratories, private companies producing antivenoms also. The scale and the size of these manufacturers vary enormously in the world. We need more manufacturers being introduced into this field, definitely. We need to improve the conditions and the capacity of the current manufacturing landscape to improve the quality and the quantity of antivenoms. So actually there is a void, a vacuum, a deficit in the supply of antivenom in the world. And for solving this deficit, we need to improve the currently existing antivenom manufacturers, the introduction of new players in the field, and the improvement of the overall quality and quantity of the antivenom field. So there are big tasks ahead of us in this particular field. This is one of the reasons why snakebite envenoming is considered a neglected tropical disease, because it has not, in addition to affecting the most impoverished populations in the world, it has not attracted the interest and the attention of the pharmaceutical industry in general. And this is something is being the international authorities are trying to change at this particular point.
And it would be safe to assume it's not a saturated space. There's definitely room.
Absolutely. For example, in the case of Africa, there is a need to increase the availability of good quality antivenoms, because in some regions of the world, there are some antivenoms being offered. Sometimes the national regulatory agencies do not keep a good control of what is being introduced in their countries, and sometimes poor quality antivenoms or antivenoms ineffective for that particular country or region are being introduced, and this is something that needs to be also solved. I would like to mention that the WHO is developing a very important program called risk assessment of antivenoms. So they open the call for antivenom manufacturers for Africa in the first step to present their products, and these are being evaluated by a committee of experts in the WHO in terms of the quality and the products and the manufacturing processes. So this risk assessment will lead to the identification of manufacturers and products that would be let's say, approved, so to speak, by the WHO, and this will clear up the landscape of antivenom availability in the world. Now, the WHO is moving to introduce this risk assessment to Asia, and in the Americas through PAHO, we are also working towards the improvement of the quality of antivenom. So there's a long way the long space for improvement in this field.
Mm-hmm. The next question is from Vasiliki Mulasiodi. She's asking, is the antivenom for veterinary use the same as the one that is for humans?
Basically, yes, just that it's formulated differently in different types of vials, and that's the main difference. But basically, it follows the same immunization scheme and the same, in our case in Costa Rica at least, and the same fractionation protocol.
So essentially, a lab that is producing antivenom is can be used potentially for both.
Exactly. There may be variations in the type of venoms used or depending on the manufacturer, but in general, the preparation is the same. It's an antibody made formulation for the treatment of animals. Mm-hmm. There are very few in the world, very few manufacturers for veterinary antivenoms.
Okay. Another question, Anastasia de Licura, she's asking, Has there ever been observed cases of evolutionary changes in the venom structure, for example, due to mutations that resulted to an antivenom becoming inefficient within its shelf life?
Yes, indeed. But these cases do not occur in short periods of time. I mean, when we see the complexity of venom composition, what we are witnessing is the product of millions of years of evolution. And it has been proposed that, for example, snake venom toxins evolve through what is called accelerated evolution. That means that the genes of the toxins are duplicated, and then these duplicated copies mutate at a high rate and generate a wide variety of proteins with different actions, compositions, and so on. And this makes the neutralization by antibodies very difficult. So this has been operating over millions of years to generate this plethora of venom variability. So it doesn't occur through short periods of time. It has occurred through millions of years of evolution. But yeah, the complexity of venoms, yeah, and the variation of venoms is due to this accelerated evolutionary process.
Which is part of what you responded, probably fits here as well. Another question reads, after the shelf life expiration date, are antivenoms still efficient? Possibly.
Yeah.
We've heard of people using them in Greece way past their shelf, the date expiration.
Well, for that question, I have an official response and then a practical response. The official response, the response that is based on the protocols accepted by ministries of health and so on, is that a liquid antivenom has a shelf life of 3 years, and then a freeze-dried antivenom of 5 years, and they should not be used after this shelf life expires. But in practical terms, we know that both liquid and freeze-dried antivenoms are effective still after this shelf life period ends. And there have been many studies showing that antivenoms are still effective after this shelf life. So in the case of an emergency and a particular situation where you have an antivenom that has expired 3-4 months ago and has acceptable, adequate appearance in terms of lacking turbidity and so on. In some cases, in particular circumstances, these have been used for treatment. So these shelf lives are established officially, formally, but we know that these antivenoms are probably more stable than we thought. This brings me to a point I mentioned in my talk, which is that antivenom manufacturers need to study the stability of their products, because a liquid antivenom may have more or less stability depending on the composition. For example, the introduction of some molecules like osmolytes improve the stability of antivenom. So a laboratory may demonstrate that a liquid antivenom has a shelf life which is more than 3 years. But this has to be demonstrated through careful stability studies for which protocols are established. And it is recommended that every manufacturer needs to demonstrate the stability of their particular products.
I just keep going with the questions, right?
Sure.
Okay.
Please.
Next question is, again, Elias Capaz. Are there consistent patterns between snake phylogenetic relatedness and venom sequence similarity?
Sometimes yes, sometimes no. It's not an automatic relationship. Sometimes you see the more distant the phylogenetic relationship, the more different the immunological properties of the venom. But sometimes you don't see that pattern. So this needs to be studied on the protein scale and on a case by case type of analysis. So
okay,
you can predict, but it does not always is the case when you do the immunological analysis of venoms.
Okay. Next question is, if hence IgY antivenom manufacture is cheaper than horse manufacture, why isn't this technology promoted to a larger scale?
Well, I am not so sure whether it is cheaper. It may seem cheaper, but you need to define the cost of the product. You need to scale the product from the lab bench type of scenario to the technological or production scenario. And sometimes there are issues that when you scale up the process, you find cost issues that you don't think of when you are working at the lab bench scale. So I wouldn't be so sure that an antivenom made of egg yolk derived antibodies is cheaper than a horse derived antivenom. This needs to be studied. By the way, it would be a very interesting research to do scale up this process and estimate the costs of production.
Another question is the production and use of a synthetic without the use of animals antivenom a viable option in the future?
The synthetic antibodies.
Yeah.
Instead of the animal derived antivenoms.
Yes.
Yeah, well, this is what I mentioned. This is a line of research that is being developed. The technologies are there. There are issues that need to be studied and solved in this particular case, but it is absolutely feasible. And in fact, several groups are trying to develop this. Whether the scaling up of these technologies for antivenom production would be realistic or effective and would bring antivenoms that would be able to neutralize complex venoms or just venoms with more simple composition. This will be defined probably in the coming years. But yeah, the possibility is there and it's absolutely realistic.
Another question from Emmanuel. He's asking, are you familiar with which are the labs that are working on veterinary related antivenom production?
Not too many. In fact, this is a very neglected field within the neglected tropical disease of snakebite envenoming. I know in Central America, we produce antivenom for veterinary use. I know in the United States, there are some antivenoms being distributed for veterinary use. I know in Brazil, there is a manufacturer for veterinary use, but I don't know of much more places where this is done. There is very little information in the literature, little information on the clinical efficacy and safety of these products. So this is definitely a field within this One Health perspective. The area of snakebite envenoming in domestic animals needs to be strengthened in terms of epidemiology, clinical manifestations, treatments, and so on.
The next question is, how many species of snakes do you have in the Instituto Clodomiro Picado, and how many animals in total? And continues by what is the amount of antivenom that is being produced from these numbers?
Okay, in Costa Rica, there are 24 species of venomous snakes. We keep in our serpentarium specimens for most of them. Some species are very scarce. Some coral snakes are very difficult to obtain. But for the species that we need to get venom for antivenom production, we have enough number of specimens in our serpentarium. I don't have in my mind exactly the number of specimens that we have, but there's plenty of snakes, especially of the ones we need for antivenom production. And then in terms of horses, we had about 120 horses in our farm at Instituto Clodomiro Picado, and to enable the manufacture of antivenoms for Latin America and Sub-Saharan Africa.
Wow, okay. And the last, previous to last question, is there a difference in antivenom quality being produced between different countries? Does it vary greatly or not?
Yes, the characteristics and quality of antivenoms greatly vary in the world. So in terms of, for example, adverse reactions in clinical trials, you see some antivenoms that induce a relatively high frequency of adverse reactions. Some antivenoms that induce very low frequency of adverse reactions. From the chemical standpoint, when you compare antivenoms, you see difference also in terms of neutralizing ability. You also see, so there are many differences. That is why this type of guideline, the WHO guideline for antivenom manufacture and control, is so important to try to standardize better the way antivenoms are manufactured. And many laboratories in the world need support in terms of training and technological transfer and so on. So we need a platform of collaboration, of cooperation, of exchanges between laboratories so that the whole landscape of antivenom manufacture can be improved on a global basis. So my idea is that this should be viewed not so much in terms of competition, but in terms of cooperation in the world.
Understood. Okay, and the last one is somebody probably watched the previous webinar. What happens in the case of hybrid snakes and their snake bites? Is the antivenom working?
Well, it depends on the hybrid. Usually hybrid snakes are relatively closely related phylogenetically. So probably it depends on which type of hybrid are you talking about. Sometimes the two parents have very different venoms, and therefore the hybrid would have like a combination of venoms. Sometimes the parents have similar venoms. So it depends on the type of hybrid you are dealing with. But I wouldn't think this would be a big problem in terms of recognition by antivenoms. It would depend on the particular case.
And the last one is, do you think there's sufficient funding going into the venom research?
Definitely not. And this is one of the reasons why this has been a neglected. Professor David Weber used to say this is the most neglected of the neglected tropical diseases. And this is true in several ways. In the sense that it affects the most impoverished population in the world, in the sense that it has not attracted the attention of pharmaceutical industry, and it has also not attracted the attention of research agencies. This, however, is being changed in the last years, thanks in part to the international initiative that allowed the positioning of snakebite envenoming as a neglected tropical disease in the WHO Department of Neglected Tropical Diseases, as a resolution adopted by the World Health Assembly in the year 2018 to urge member states to take care of snakebite envenoming. So after that, new opportunities are emerging for research. In fact, for example, the Wellcome Trust in the UK is approved a program, initiated a program with 80 million pounds to support different projects to improve the research and development and technological development in the field of antivenoms and snakebite envenoming, and several other foundations, like the Lillian Lincoln Foundation, the Hamish Oxton Foundation in the UK, and other foundations are also introducing funding for this particular field. So things are changing, and now there are more possibilities to get funding for research and development in this field, and hopefully this will increase in the future. So thanks to this international initiative, things are improving significantly in our field.
Okay, and the last question is, I would assume it's the last question. Is there any variability in symptoms after a snake bite from the same species?
Yes. For example, let me give you an example. Well, one good example is, again, the Russell's viper in the Indian subcontinent. There are some populations of Russell, same species, the Boya russelli, some populations which inflict an envenoming characterized by bleeding, local tissue damage, coagulopathies, for example. Other populations have induced clinical manifestations with the predominance of acute kidney injury, severe damage in the kidney, which occurs in a high percentage of the cases. In other cases, there is a clinical manifestation characterized by what is called systemic capillary leakage syndrome, which induces an increase in plasma permeability in different parts of the body. In other regions, there are venoms from Russell's viper that inflict neuromuscular paralysis, for example. And there is a whole combination of these manifestations. So it's a big challenge clinically. This is probably one of the most clear examples of clinical variation in the manifestations from a single species, but something similar occurs in other species as well. In contrast, for example, in Latin America, envenoming by Bothrops specimens are usually have the same clinical manifestations of local tissue damage, systemic bleeding, coagulopathy. However, one case which is interesting are two species endemic to the Caribbean islands of St. Lucia and Martinique, Bothrops lanceolatus and Bothrops caribeus, which inflict a thrombotic type of effect, which is not seen in other Bothrops snakes. So, as I mentioned, the variation in the biochemical composition translates into variation in clinical manifestations That's a big challenge for antivenom manufacturers and for clinicians treating the snake bite.
Yes, and I think we had this discussion with David Worrell. Essentially, we're discussing for a Greek species, which is the Vipera ammodytis, and then we have the same species in Croatia, and there's different symptomology. Essentially, trying to see what's going on with that. I think this essentially concludes all the questions. Quite a lot of questions. So at this point, I think the presentation is also concluded. Officially, we would like to thank you very much for taking the time and being with us. Again, taking the time to answer all these questions. And thanks to everyone for attending. A lot of very nice comments here. Thanks to everyone. You're very
Thank
much
you very
welcome.
much. Thank you very much, Thomas, and thank you very much to all the people that attended this activity. I felt very honored to be able to attend this seminar, and I really appreciate the invitation and all the questions and the interest that people showed.
Greetings to Panama. We have.
Okay, Ildaura. Nice to see you, Ildaura, here. Thank you very much for being present.
I think we have people, Cyprus, Spain, France, Costa Rica, well, Panama, Greece. Yes, of course, a lot of people here. The United Kingdom.
Venezuela.
Right.
Great.
Venezuela. We also have with us a professor of biochemistry, Maria Turaki. Thank you for being with us. Venezuela. Wow. Okay.
Fantastic.
Yeah. So, yeah, I think at this point, this essentially concludes it. Of course, we can chat, but all the attendees can feel free to leave and whatnot. Thank you very much for being with us and for supporting this activity.
Thank you.
Is David with us? I would assume.
Yes, I'm still here.
Hi.
Well done, Chairman. That was a very beautiful, very clear talk, and you dealt with the questions in a masterful manner. And congratulations to Thomas's audience for an interesting range of questions. Pity there wasn't more time to pursue some of the lines of inquiry.
Well, especially for the hybrid species. I remember, that's why I said somebody probably attended the previous one. You had mentioned hybrid snakes and how difficult managing such a snake bite is.
Well, I think it's a potential problem, clinical problem, but at least usually the victim of a hybrid snake bite knows the nature of the hybridization. Mm-hmm. And as Chema said, hybridization is most likely to be between fairly closely related species. Of course, the definition of a species used to be that hybridization produced fertile offspring, but species are now a much more vague concept, really, based on phylogenetic distance and so on. But it's an intriguing question, but I agree with Gemma that it's unlikely to be a practical one.
Mm-hmm.
But it's interesting, the question came up about IgY antibodies. I think there is a serious question about the affinity of IgY antibodies. So quite apart from the difficulty that Chema mentioned about scaling up production, I think there may be a more fundamental problem about the structure of IgY antibodies and whether they neutralize venom toxins adequately. Mm-hmm. Mm-hmm. I mean, that was an idea that was applied to veterinary antivenoms in Australia in many years ago. And in fact, there was a commercial product available, an IgY antivenom for veterinary use in Australia, but that initiative was not sustained.
Mm-hmm. At this point, I will say if anyone from the audience, some people are still here with us, they want to speak, they can just write it and I can just give them the privileges since Q&A was done in an efficient manner. so you can just text me. What I also like to ask, since you're here with us, is clinical. I mean, what's going on is captive collections have started hybridizing wide variety of animals. What happens if someone gets bit? I mean, we could take, for example, species that are not very closely related, and apparently they give viable offspring. I mean, I've seen hybridizations within the Vipera and then other genuses, which is just out of this world. What happens there? The clinical management side of everything.
Well, we wait and see. I could dream up some very difficult clinical scenarios, but I don't know whether it's possible yet. I mean, the purpose of this hybridization is usually to produce more attractive color varieties, isn't it?
Yes, absolutely.
And so on. I mean, I feel, Thomas, very much that Dr. Gutierrez has been speaking now for nearly 2 hours. I think we should respect his, although he's a very young man with boundless energy, I think we should probably let him off the hook at this stage. But I mean, it's such a fascinating subject. One could go on chatting about it for hours. And there were many things that came up in the talk. For example, the window of opportunity, the therapeutic window of opportunity for antivenom treatment. We could talk more about that. And it's a fascinating subject. And of course, Dr. Gutierrez has been one of the great pioneers, not only of antivenom production with the Instituto Clodomiro Picado, but also these visionary ideas about ancillary treatments like small molecular weight enzyme inhibitors and so on. So I mean, there's a lot more that could be discussed. I think you should try and persuade him to come back again in the future.
Yes.
Anyway, thank you very much for letting me join in, Thomas. And
Absolutely.
Always a great pleasure to see Jose Maria Gutierrez and to hear his wisdom and to sit at his feet, as it were.
Likewise, David. Likewise, it's always a pleasure to see you with your brightness and your whole perspective of all these problems. So it is an honor to share this session with you and with Thomas and all the people that attended, of course.
Anyway, goodbye. Good luck. Stay healthy.
Take care, David.
Take care.
Very nice to see you all.
Likewise.
Yes.
Okay, so at this point, I think I will end the entire webinar.
Okay.
Just like my professor essentially mentioned, the series should continue. At some point, yes, I think the clinical management side of things, that is definitely something that a lot of doctors have come up and said, we would like to know more. We have some of them today here with us, but for the future, let's take a break now.
Yeah, and another possibility for the future would be to bring this topic of new alternatives, the recombinant antibodies and the inhibitors, which is quite exciting because we have already clinical trials going on inhibitors for the first time in history. So
they have begun.
Yeah. One in Kenya and one in the USA and India.
So far, it's looking promising, I would assume.
Yeah, it's looking promising from the preclinical. We have done the preclinical studies, and it's very promising. So I think that would be another interesting topic for your audience, Thomas.
Absolutely. Okay, so thanks to everyone for attending. Thanks to you, Senor Gutierrez. We're forever indebted to your debt. So, yeah.
Thank you very much.
We will talk anon.
Sure. Take care, everybody, and thanks for the invitation. It was a great pleasure.
Our pleasure to have you with us.
Bye-bye.
Bye-bye.